Our next speaker is Leslie Berg, Professor Leslie Berg, who is the chair of the Department of Immunology at the University of Colorado, Anschutz. She's also a former president of the American Association of Immunology. And her scientific interest is immune cell signaling and T cell signaling. And this stems, I think, from the time in her lab as a postdoc in the lab of Mark Davis at Stanford, where she carried out seminal studies on T cell development through the ingenious use of MHC class II restricted TCR transgenic mice. And she has continued this research at the forefront really of T cell signaling mechanisms and unraveled, for instance, the roles of tyrosine kinases in T cell mediated immunity and signaling. So we are really delighted to have an expert on T cell signaling to introduce us to key aspects of immune cell signaling. Leslie, welcome to.
Great, thank you, Stefan. So is my presentation showing?
Yes, it is.
Excellent, okay. And thank you to Hida for doing such an outstanding job introducing all the basic elements of the immune response. So we're gonna focus in my hour or 50 minutes on signaling pathways. And even though I actually, this talk was titled Immune Cells, I think really I should have called it lymphocytes, because really focusing on lymphocyte signaling pathways. So first I was gonna start with a very brief introduction to how pathogens meet lymphocytes and lymph nodes. Hida outlined that, so I won't belabor it I'll try to go through it pretty quickly. So the anatomy of the immune response is how I think of this. The second is talking about the major modules in the biochemistry of antigen receptor signaling and their functions in activation of lymphocytes. And this involves a discussion of kinases, adapters, and then second messengers. And of course, we'll briefly cover the pathway leading to the calcium response, which Hida already, as well as Dr. Long, already introduced. And then finally, the last part, I was going to talk about the modulators of antigen receptor signaling, because these have become more and more prominent, I think, in recent years in immunology. These are what we think of as the co-stimulatory pathways, as well as the inhibitory receptors that are often induced and upregulated after activation of lymphocytes. And so these modulators really you could think of as the receptors on lymphocytes that help the T or B cell figure out what's going on in its environment. So antigen might be present, but is there really an infection? And if there's an infection, when is it gone? So if you clear a virus infection, as Hida showed you in his kinetic curves, you want your immune system to shut off and stop responding once the infection's been cleared. So that's really one of the major functions of these, what we call co-stimulatory and inhibitory pathways. Okay, so Hida already covered this, that the immune system is divided into these two sort of temporally distinct waves of responses: the innate immune response, which happens locally right at the site of an infection. When the barrier is breached, bacteria, viruses, fungi, whatever get in, and that's induces this very rapid response at the site of infection. And then as that innate immune response ensues, it elicits an adaptive immune response, assuming that the innate immune response is ineffective at eliminating the pathogen. And that adaptive immune response, as Hida mentioned, is very slow. It's incredibly specific to the specific pathogen that you've been infected with, but also, as Hida also mentioned, it is initiated not at the site of infection where the innate response occurs, but in these centralized structures in your body that are the collecting sites for antigens, pathogens, or toxins, or whatever else from around the body. And these are your secondary lymphoid organs, all of them shown in yellow here. So lymph nodes, spleen, adenoids, and so on. So again, as Hida originally told you about, your T cells and B cells, we think of the cells when they're in the state before they've ever been exposed to an antigen or pathogen. So they finish their development, B cells in the bone marrow, T cells in an organ called the thymus. They exit those tissues and become circulating lymphocytes. We call them naive if they have developed but never before been activated by encountering their specific antigen or pathogen. And those cells have this sort of trafficking pattern where they go in the blood. At some point, they exit into a particular lymph node for no particular reason that we know of. They sample each lymph node. And if they don't find their specific antigen, so in a healthy person, these cells are recirculating. They look in the lymph node for their antigen. If they don't find it, they'll leave via the efferent lymphatics, and they eventually drain via the thoracic duct back into the circulation. And so these recirculating T and B cells will end up sampling all of the different lymph nodes and secondary lymphoid organs in your body. As it's been estimated, roughly each cell will sample each lymph node roughly once a day. So they're essentially on patrol in your circulation and in your secondary lymphoid organs looking for infections. So what happens, as again Hida outlined, is that in all of our tissues, living in those tissues are these sentinel cells we call dendritic cells, and their job is to sense barrier breaches via an entry of pathogens, infections, and so on, and to get activated, which elicits both a change in the behavior of the cells. So they now, instead of remaining resident in the tissue, they will pick up and move into the draining lymphatic fluid from that tissue and drain into the nearest lymph node that's essentially downstream of these lymphatic system. So that's why people refer to the draining lymph node as the site where you can find evidence of infection. So if you have strep throat, the doctor will start touching your neck and looking for enlarged lymph nodes. And so these are the draining lymph nodes. The dendritic cells will carry the antigens to those draining lymph nodes where they can encounter these recirculating B and T cells. So just to kind of put all of this back into context, we have a breach in the barrier or an infection of a virus through the epithelium, some epithelial surface like the epithelium of your lung, for example. And that virus will be taken up by the tissue-resident dendritic cells in that tissue. The dendritic cells will pick up and take that tissue into a lymphatic. It will activate the dendritic cell, take it into a lymphatic vessel, draining it into a lymph node, the draining lymph node, where your recirculating B cells and T cells will encounter antigen. If they're specific for that antigen, they'll get activated. And then those activated T cells and some B cells, but largely the antibodies made by the B cells, as Hida outlined, will traffic back to the site of infection through the circulation and leave the blood vessels at the site of infection and go out and try to, eliminate the pathogen. Okay, so that's the anatomy. And now we're going to focus on what happens in that lymph node when to activate the lymphocytes. So I like to always start with this picture. It's one of my absolute favorite pictures. I guess Hida had his favorites too, but this is my favorite picture. It's a series of transmission EMs from Jim Eiley's lab a number of years ago, where they took a naive resting T cell and imaged it, and then activated that T cell by stimulating the T cell receptor on its surface, and then took pictures at these different intervals. And what I think you can see, and it's kind of interesting when we heard Dr. Long present this interesting physical interaction between the STIM1 and 2 proteins and the ORI1 channel and the plasma membrane. It sounds really outrageous to imagine an ER resident, membrane resident protein touching a protein in the plasma membrane. But look at the naive T cell. This black is the nucleus, right? And this is the plasma membrane. So naive T, and this is 2 microns. so naive T cells are unbelievably small, and they are, I don't know, 90% or so nucleus, have very, very little cytoplasm. And so in fact, they're really, maybe it's not such a stretch that the ER could, come in close proximity to the plasma membrane. And if you look over the course of half an hour and 1 hour and so on, what you start seeing is the changes in the T cell. The chromatin decondenses, the cell enters the cell cycle, the cell upregulates mechanisms to start synthesizing. If you imagine, this is what it starts with. And in 48 hours, it looks like this. Somewhere between these two last time points is when the T cell will undergo its first cell division, and then it will start dividing, if it's a cytotoxic T cell, once every 4 to 6 hours. So in 4 to 6 hours, over and over and over again, the T cell has to resynthesize every macromolecule it needs to double its content of ribosomes, lipids, membrane, other membranes, proteins, everything, DNA, you name it, in order to divide at that rapid pace. So this cell is a completely different beast than the one we started with, and that's really the job of all these activation pathways that we'll talk about. Okay, so how has all this happened? This is the membrane-bound form of the immunoglobulin protein. Again, you heard about from Hida, and this is the similar version of the T cell antigen receptor. It's both of these proteins, the antigen recognition domains, of course, are the distal part away from the cell membrane. And these proteins themselves don't have really any signaling function. You can see these tiny little cytoplasmic tails in the membrane-bound IgM and the T cell antigen receptor, 5 amino acids, I think each one is. So there's nothing that receptor can do in terms of signaling. And so the way these receptors signal activation or antigen binding is by being associated with transmembrane proteins that are the signaling components. So for the B cell receptor, it's these this dimer called Ig alpha, Ig beta. For the T cell receptor, there are actually several different signaling proteins. Collectively, they're called the CD3 proteins. And then there's a separate sort of structurally distinct signaling protein associated called the T cell receptor zeta chain. And all of these signaling proteins share these motifs in their amino acid sequence in their cytoplasmic tails that are called ITAMs. It's a crazy acronym. It's essentially a tyrosine motif that is phosphorylated as the first step of T cell receptor or B cell receptor signaling. So here's a picture of the T cell receptor. This is the sequence of an ITAM. So there's 2 tyrosines in each ITAM, little consensus sequence set, and they're separated by about 10 or so, 11 amino acids. And what happens as the first step of signaling is that these ITAM sequences are phosphorylated on both tyrosines, and that recruits a kinase that has a dual or what's called tandem SH2 domain. So this kinase will not bind if only one tyrosine is phosphorylated. It requires both tyrosines to be phosphorylated in the same ITAM. So for the T cell receptor and B cell receptor, the first step of signaling is the activation of tyrosine kinases, which first phosphorylate those ITAM motifs in these cytoplasmic tails of the signaling domains. And then we're going to talk about some of the players in these various steps that come next, but after activating tyrosine kinases, those kinases phosphorylate proteins called adapter proteins, which we'll talk about 1 or 2, and they essentially are Velcro that just basically bring signaling proteins together at the plasma membrane in order to promote the downstream signaling pathways. The adapter proteins recruit and activate both phospholipases and lipid kinases to elicit second messengers that are often associated with the activation of small GTPases, a whole plethora of different serine-threonine kinases, and phosphatases like calcineurin that is, of course, important and responsive to calcium signaling. And so these are the real effectors of the signaling pathways that are individually responsible for promoting different functional consequences in the activated lymphocyte. And then as a result of these effector functions, you activate transcription factors to turn on new gene expression in the activated T or B cell. You induce a bunch of cytoskeletal changes, and you dramatically change the metabolism of the cell. And that those are just a few of the changes that occur as a result of these antigen receptor signaling pathways. So here we have the T cell receptor with its associated first kinase that's activated. This is a Sark family kinase, LCK. That LCK kinase phosphorylates ITAM motifs in the CD3 and zeta protein. That recruits a protein in T cells called ZAP70, which simply is an acronym for zeta associated protein 70 of 70 kilodaltons. And this ZAP70 protein then phosphorylates adapter proteins that lead to forming signaling complexes that then do the sort of business end of changing the biology of the T or B cell. And B cells have a very similar organization, although the proteins in the B cell are distinct from those in the T cell. They don't share that many signaling proteins, but they share the overall logic of how the signaling is mediated. So for B cells, there's a Sark kinase called LYN. It phosphorylates the ITAMs in Ig alpha and Ig beta. That recruits a protein that's a family member of ZAP70. In B cells, that protein is called SYK, and SYK as well activates phosphorylates adapter proteins, leading to forming signaling complexes. Okay, so as I mentioned, these downstream signaling events are really dependent on the phosphorylation of adapter proteins. So probably most of you are familiar with adapters, but just in case you're not, I think of them as proteins whose function is to essentially serve as protein-specific Velcro for binding partners. So some adapters will bind to a protein in the membrane that will help them localize to the membrane. They can, because they're phosphorylated, the adapter proteins, they will recruit proteins in a reversible manner. So when the adapter is phosphorylated, a protein will bind. When the adapter gets dephosphorylated, that protein will fall off. So this is a way to have reversible signaling processes occur. You can also have adapters serve as kind of scaffolds to bring enzymes together with their substrates. And also in some cases, binding of a protein to an adapter will lead to a conformational change in the bound protein that affects its activity. So that's the overview. And in T cells, the main adapter protein that serves this kind of big scaffolding function is the protein called LAT, which stands for linker of activated T cells. And in B cells, there's a similar protein that's called SLP65. And so these proteins each are phosphorylated by this kinase with the tandem SH2 domain. In T cells, ZAP70 phosphorylates LAT on at least 4 and possibly more tyrosines. Similar, and each of those tyrosines recruits different proteins because they're embedded in different amino acid sequences. So they recruit sometimes enzymes or GTPase GEF proteins. They recruit other adapters. They can recruit enzymes like phospholipase C gamma, which is a key mediator of T cell receptor signaling. And B cells, similarly, this adapter protein gets phosphorylated by SYK, the tandem SH2 domain containing kinase in B cells, and that to phosphorylated adapter recruits other GEF proteins, BTK and other tyrosine kinase, some more little small adapters, and also recruits PLC gamma, which plays a role both in B cell activation as well as T cell activation. Okay, so two of the pathways that are activated in both B cells and T cells by this process lead to the production of active small GTPases. So in T cells, these adapter proteins recruit a GEF protein for CDC42. And when CDC42 is activated, it leads to the polymerization of actin, which is a key step in changing the cytoskeletal organization in the lymphocyte. A second protein that's recruited to the same adapter complex has the ability to lead to the activation of another small GTPase, RAP1, which if I think I am correct, it's a member of the Rho family of GTPases. And this one leads to the activation and clustering of integrin, which are basically cell adhesion proteins on the surface of the T cells or B cells, which allow them to bind more, adhere more tightly, in the case of T cells, to the dendritic cell that's activating them. So these are two of the effector functions that are induced by this whole process of kinases phosphorylating adapters. And to summarize these points that we've covered so far, antigen receptor signaling pathways are initiated by activation of tyrosine kinases. First, we have Sarc family, then Syk family, and then there's a third family of tyrosine kinases. I mentioned BTK in B cells, and there's a family member also in T cells. The Syk and ZAP70 kinases phosphorylate adapters and scaffolds, and that leads to activation of an enzyme called PI3 kinase, which is a lipid kinase, modifies PIP2 in the plasma membrane of the lymphocyte that GEFs for the small GTPases I talked about, and it also ends up leading to PLC gamma activation. And the outcome of these pathways is a whole bunch of changes. You saw the image of what happens to a naive T cell when it becomes an activated T cell. And so the functions that are altered in the T cell during that process are enhanced survival, upregulated metabolism, macromolecular synthesis. The cell acquires the ability to migrate. It has reorganized its actin cytoskeleton. It's got better adhesion properties, and of course, activation of transcription factors that will lead to new gene expression. Okay, so I kind of break down this whole series of pathways downstream of the antigen receptors into these three modules. PI3 kinase is activated by both B and T cell receptor signaling. And without going through all the details, this is the pathway that's really essential for both enhanced survival of lymphocytes when they're activated, as well as their altered metabolism, and most importantly, their greatly upregulated ability to synthesize their contents. Like they upregulate protein synthesis machinery. They have upregulate glucose transporters so that they're better at taking up glucose to make energy for all of the things that the cell's going to have to do, and so on. The second big module is the activation of GTP exchange factors, which lead to activation of these small GTPases, which, as I mentioned, are important for actin cytoskeletal reorganization, adhesion, and also play a role in cell migration. And the third branch is the one that was originally probably studied the best, and is possibly the most well understood, and that is the activation of PLC gamma 1 in T cells and also in B cells. And this phospholipase, as Hida already introduced you to, is responsible for cleaving PIP2 in the membrane, plasma membrane of the lymphocyte, and generating these two second messengers, diacylglycerol and IP3. And we heard, of course, a wonderful talk from Dr. Long about the function of IP3 binding to the IP3 receptor and how that leads to calcium influx, which I'll also briefly show you the pathway for. And the other second messenger, diacylglycerol, is really important in activating other kinases in T cells, protein kinase C, which leads to NF-kappa B activation, and the ERK pathway, which leads to the production of another transcription factor called AP1. Okay, so here's how PLC gamma gets activated. We have LCK, ZAP70, adapter complexes, and I left out all the little proteins involved that recruits this third tyrosine kinase. So in B cells, that protein is BTK. In T cells, it's a relative of BTK called ITK. And ITK and BTK are the kinases, tyrosine kinases, that actually directly phosphorylate PLC gamma 1 to activate it. And the active PLC gamma 1 then cleaves PIP2 to generate the membrane-associated second messenger diacylglycerol and the soluble IP3. And these, in turn, activate three major transcription factor branches. Protein kinase C, again, activates NF-kappa B through many steps, the RAS-MAP kinase pathway, and then calcium influx. And those three sort of transcription factor pathways then lead to transcription factors that can enter the nucleus and bind to DNA and elicit transcription. Okay, so here's PLC gamma cleaving PIP2 into these two second messengers. So diacylglycerol remains associated with the plasma membrane, activates these pathways shown here. And then we have the soluble IP3 that binds to IP3 receptor. Unfortunately, this blob here probably doesn't resemble in any way, shape, or form the beautiful structure that we saw, but it causes the opening of this calcium channel, allowing calcium to leave the endoplasmic reticulum. And so as a result of IP3 binding to this IP3 receptor and calcium coming out of the ER into the cytoplasm, as we heard, there's a STIM1-2 complex in the ER membrane, which is normally associated with calcium. And when the levels of calcium inside the ER drop sufficiently, the calcium ions fall off STIM1 and STIM2, and that elicits a conformational change in that complex, which causes both aggregation and probably extension of the STIM1, STIM2 complex, allowing it to interact with this ORI1 calcium channel on the plasma membrane. And so this is the mechanism which immunologists had discovered, I don't know, decades ago, that was why this protein was called calcium release activated calcium channel, was because it was known that calcium came out of the ER, and it was thought initially that that calcium was directly activating this crack channel, but in fact, it's not a direct effect. It's an effect of the loss of calcium from the ER that changes the binding ability and conformation of STIM1-2, allowing it to activate ORI1. Okay, so just in summary, we have these three major signaling molecules. They have important functions, and we can account for, many of the changes that take place in the T cell upon activation through these different signaling pathways. Okay, so modulators of antigen receptor signaling. So now things get a little bit more complicated. If it was just up to the T cell receptor and B cell receptor, I think we feel we have a reasonable handle on how all that works. But it turns out that there are multiple receptors on the surface of lymphocytes, both B and T cells, that change the signaling that might activate the cell through the antigen receptor. So these receptors, they're often called co-stimulatory or inhibitory receptors, very often, and for the most part, have no function when they're engaged on their own. So they don't actually have an ability to change the function or outcomes of a B cell or T cell when stimulated all by themselves. But what they do is they change the response inside the T cell or B cell to the stimulation or activation of their antigen receptor. And that's why we think of them as modulators of antigen receptor signaling. And so there are both co-stimulatory receptors that enhance B and T cell activation. And for T cells, I think many of you may have heard about things like CAR T cells, which are really prominent right now in efforts to design or develop better means for treating tumors. So this stands for chimeric antigen receptor. And this is, these are made by using a single chain FV region of an immunoglobulin protein as a binding domain on a chimeric receptor. And those single chain immunoglobulins are designed to bind to a protein expressed on a tumor. One of the common ones targets some B cell lymphomas by having a binding domain to the B cell lymphoma. And then in order to elicit a function out of the T cell, in most cases, these are engineered into cytotoxic T cells to cause them to bind to a tumor and kill the tumor cell. They've engineered in their cytoplasmic tail signaling domains stolen from the T cell receptor as well as a co-stimulatory receptor. So two common versions of these CAR or chimeric antigen receptors against tumors have the zeta chain of the T cell receptor with the ITAMs that are phosphorylated, recruit SAP70 to initiate signaling, and a piece of a co-stimulatory receptor, either CD28 that we'll briefly talk about, or a piece of a TNF receptor that's called 41BB. But in addition to co-stimulatory receptors, there are inhibitory receptors. And these are also now heavily in the news in terms of cancer therapy. These are the targets of checkpoint blockade therapies. So these receptors suppress T or B cell activation. And so when you have upregulation of an inhibitory receptor, let's say on a T cell, now when that T cell finds its antigen, let's say on a tumor cell, it's not going to be able to kill the tumor cell because it's got inhibitory receptors that are engaged that are going to shut off the activation of the T cell. And so checkpoint blockade treatments are refer to giving patients antibodies that block these inhibitory receptors. So now when those T cells encounter the tumor cell and their T cell receptor is stimulated, there's no inhibitory receptor engagement with its ligand to try to turn off the T cell. And these are the treatments that have been so effective in allowing people's own T cells to do a better job of killing tumor cells in cases of some forms of lung cancer and melanoma and other things. So how do they work? So before we get into a little bit of the signaling, I just want to try to put this in context. So I personally have always had a hard time trying to figure out why T cells would upregulate receptors that turn them off. You know, you're trying to make an immune response. So I get co-stimulatory receptors. So they are expressed and they help enhance T cell or B cell activation. But inhibitory receptors are upregulated shortly after, in the case of T cells are activated, and their job is to turn off the T cell response. So why would you do that? Well, as Hida kind of alluded to, you need really to tightly control your immune response. So not only do we need to maintain self-tolerance when there's no infection, we want to make sure that our lymphocytes are only activated when there really is an infection. So that's one of the functions of these modulatory receptors. The other thing to think about is that the immune response should be right-sized. So what does that mean? So it means that if the infection is a huge titer of bacteria in your tissue, you've got this festering, horrible, infected wound, you want to make a bigger immune response, adaptive immune response, than if you've just, got a little cut in your finger and, 10 or 20 or whatever, bacteria get in. So, and the reason for that is as you can probably guess, your immune cells your lymphocytes, which then recruit all of these innate cells like macrophages and neutrophils, can do a huge amount of tissue damage. And so we really don't want to overdo the immune response if it's not necessary, because there's a lot of collateral tissue damage when you make an immune response. And secondly, you also want to know when that pathogen or antigen is gone. So once you've eliminated the virus, you want to get rid of those antigen-specific responses because it's over and done with. And again, the immune response can cause tissue damage, and we want to get that over with when the pathogen has been cleared. And that's really the job of these co-stimulatory and inhibitory receptors, is to help modulate the response of the lymphocytes. Okay, so a balanced immune response, of course, as you already heard, is you get a virus infection. If it's something that's not dealt with by your innate immune system, you initiate an adaptive immune system. I'm just showing T cells here, not the B cell response and antibody production. But usually after about a week to 10 days, you start having a T cell response that usually does a good job in getting rid of the virus. And then you want those T cells to disappear for the most part, leaving a few behind to give you some long-term memory, but mostly getting rid of those T cells. Most of the majority, people have estimated maybe 90% of them die at the end of an immune response when the antigen or pathogen has been cleared. And so this is all over with, and by the end you keep a few around, maybe 10% of the ones you had at the peak over here, and these are what provide this memory response that Hida mentioned in the case of T cells. But so here's the infection, the response, and then this back to homeostasis, which requires that lymphocytes die. You generate some quiescent memory cells that look a lot like those naive T cells. They go back to being very small and metabolically quiet. And you also have to repair any tissue damage that the immune response has caused. So when that process is unbalanced, for example, if your immune system doesn't do a good job in clearing the pathogen, you can get chronic infections or cancer that can't be eliminated by your immune system. And if the immune response is too big and doesn't stop when it's supposed to, you end up with chronic inflammation, a lot of tissue damage, and can even end up with autoimmune diseases. So it's important that this immune response be balanced. And that is really why your immune system not only has ways to activate lymphocytes, but also ways to help shut them off. Okay, so antigen receptor signals are balanced by this combination of activating and inhibitory receptors. And one thing we're really just going to gloss over is that there are multitudes of these receptors. So this shows you one co-stimulatory receptor, CD28, one inhibitory receptor, PD1, but there are actually dozens of them. I think I have a slide just showing you how kind of overwhelming it is. And they're expressed with different kinetics in T cells. They're expressed on different types of T cells and B cells. They differ in whether they're upregulated by different cytokines or growth factors in response to something happening in the environment. And importantly, the ligands for these receptors are expressed on different cell types. So the receptors, both the co-stimulatory and the inhibitory receptors, they're not interchangeable. So they're going to be engaged and therefore induce signaling in different contexts and different tissues. Okay, so how do co-stimulatory receptors work? So here we have B cell receptor or T cell receptor activating a lymphocyte, and these responses can be enhanced by co-stimulatory receptors. So in T cells, a main co-stimulatory receptor is CD28. In B cells, CD19 is also very important, as we'll see in a minute. And in addition, there are members of the TNF receptor superfamily that also function as co-stimulatory receptors in B and T cells. And these co-stimulatory receptors basically activate PI3 kinase. So they play a role in enhanced survival and altered metabolism in the B and T cell. And they also help activate transcription factors, in particular NF-kappa B. So here's the case of a B cell, and thankfully HIDA introduced this idea about complement. So if we have a bacterial infection, the bacteria have structures on their surface that will be recognized by antibodies specific to that bacterium. But as Hida mentioned, these bacteria, usually as soon as they enter your body, they're going to start getting decorated with complement proteins on them, because there's complement proteins all around in your body and your tissues and your blood all over the place. And they will recognize aspects of the bacteria and covalently attach complement proteins to the surface of the bacteria. So if we have a B cell that's recognizing, has some specificity for a surface structure on the bacterium, and that bacterium is also decorated with complement proteins, the complement will engage a receptor called the complement receptor, CR2, which is associated with this co-stimulatory receptor CD19. And what happens is now you bring the co-stimulatory receptor, or CD19, just together in proximity with the B cell receptor. And there's a Sarc family kinase sitting here that will phosphorylate the ITAMs on the signaling subunits of the B cell receptor. And the consequence of this sort of dual engagement of both the B cell receptor and the complement receptor is that this B cell is now 100 to 1,000 times more sensitive. So when there's only small numbers of bacteria around, maybe they're not enough to stimulate or activate a particular B cell. But if the complement receptor also is engaged, now this B cell will get activated by, again, orders of magnitude lower concentrations of antigen. And so it's a way to dramatically enhance the sensitivity of the B cell response by this dual engagement of both the B cell receptor and the co-stimulatory receptor. So how they work is that both CD19 on B cells or CD28 on T cells have another motif in their cytoplasmic signaling tail that gets tyrosine phosphorylated, and that can recruit PI3 kinase. And when that PI3 kinase is recruited, it's activated, and it phosphorylates lipids in the membrane to create PIP3, which will then activate things like AKT, mTOR, and also lead to the recruitment of phospholipase C gamma. So this is a means of just enhancing signaling that's already being induced by the antigen receptor, but now you enhance the efficiency of that process. And a second kind of co-stimulatory receptor are members of the TNF receptor family. I'm not going to go through all these details, but just like the T cell receptor has an ability to activate NF-kappa B, one of the main downstream responses to engaging TNF receptors is that they activate NF-kappa B. And so this, again, just greatly amplifies the response of the T cell receptor or B cell receptor to whatever is stimulating it. So co-stimulatory receptors enhance lymphocyte activation. They can increase sensitivity to low levels of antigen, promote lymphocyte survival, increase cellular metabolism, macromolecular synthesis through mTOR and AKT, and their ligands are generally upregulated by the innate immune response. So that helps the lymphocyte decide, yes, this is really a danger, there's an infection, there's inflammation around me, and so I'm going to, increase my sensitivity to respond to whatever that pathogen is. So this is from years ago, and maybe there are even more of them now, a list of a whole bunch of these co-stimulatory and inhibitory receptors, just the ones on T cells. And so these are the CD28 protein family members. There's both activating as well as inhibitory receptors in that family. Right here is a whole plethora of these TNF receptor family members. So there's enormous numbers of people trying to figure out all the time what the function of all of these different receptors, both the activating and the inhibitory receptors on T cells are, and when they're expressed and what their functions are in terms of regulating immune responses. Okay, so inhibitory receptors in the last couple minutes suppress T or B cell activation. In T cells, there are a couple that are well known, CTLA4 and PD-1. Both of these are proteins for which there are blocking antibodies that are used in patients as checkpoint blockade reagents to try to enhance the patient's own T cell response to a tumor by blocking these inhibitory receptors. They're also important not in everybody, not just when someone has cancer, because they're important in maintaining tolerance, self-tolerance. In B cells, we have a receptor to Fc gamma, the Fc portion of immunoglobulins. There's another inhibitory receptor that binds sialic acid. And the general thinking about these inhibitory receptors is that if there's already antigen, a pathogen bound to antibody, you want to shut off your B cell because it's not needed. There's already other B cells responding, making antibody. So this receptor binds antigen antibody complexes and shuts off the B cell. Sialic acid, of course, is at high levels on most mammalian cells. So this is a way of your B cells kind of saying, no, maybe this is not a pathogen. Maybe this is actually just kind of weird, human cell. And I don't really want to make an antibody to that. So this sialic acid binding receptor is also important in shutting off B cells. So signaling wise, how they do this is by activating either protein or lipid phosphatases, which of course reverse all the kinases that are activated. The activity of the kinase is activated by the antigen receptor. And just as one example, here's PD-1, one of the inhibitory receptors on T cells. And when PD-1 is engaged, it leads to phosphorylation of its cytoplasmic tail, which recruits a tyrosine phosphatase called SGP2. That tyrosine phosphatase undoes all the phosphorylation events that are stimulated by engaging the T cell receptor. So it undoes the phosphorylation of CD3, all the other signaling components, and of the co-stimulatory receptor CD28. And so as a result of that, it essentially turns down the response of the T cell. So we have these three signaling modules that I went through. I'm not going to go back through the details, but starts with antigen-specific activation, and the activation, as we went through, is enhanced by co-stimulatory receptors that are sensitive to sensors of infection, and it's inhibited. These pathways are inhibited or suppressed by inhibitory receptors, whose function is really to prevent T cells becoming a cause for autoimmunity, or B cells and antibodies becoming a cause for autoimmunity, as well as to prevent overexuberant immune responses and to help shut off the response once the pathogen has been cleared. And now I'm finished. I'd be happy to take any questions.
Thank you, Leslie. That was a very clear lecture again. And maybe I can get started with one question by Mira Yul Kazi. And I guess the three signaling pathways that you described downstream of the T cell receptor, it's well known that they're involved in the production of cytokines that are involved in immune activation or T cell mediated immune responses. But the specific question here is, are these signaling pathways also involved in the production of cytokines that are involved in the resolution of inflammation, basically that have anti-inflammatory effects?
Yes, absolutely. So I think that the answer to that is mostly T cells. I think B cells are not considered major producers of cytokines, but T cells certainly make cytokines, and depending on the timing and the infection and the flavor of T cell, they can make cytokines that will actually suppress macrophage activation to turn off inflammatory responses. There are also subsets of T cells whose only job is to suppress immune responses. So when many of the T cells that live in your intestinal epithelium or in epithelial tissue, their job is to prevent your immune system from reacting to your commensal microbes in your gut, and to prevent your T cells from reacting to food antigens. So those are actually resident T cells in the gut that make a variety of anti-inflammatory or sort of suppressive cytokines and have the ability to shut off and dendritic cells, shut off macrophages, and shut off other T cells.
Hey, Stefan, can I interrupt and I ask a question if you don't mind?
Sure, go ahead.
Leslie, so I was really fascinated and I was thinking about...
Did you say your name?
Sorry, this is Bimal. I'm one of the organizers.
There, I couldn't see you.
Okay. Yeah, sorry, my video was off. So I was wondering, with all the heterogeneity that we see in T cells, in some ways, the immunologists were probably ahead of their time looking at all the single cell and like the lot of heterogeneous subsets. How much heterogeneity do we see in the signaling frameworks within these different T cell subsets?
Very little. I think what changes is when T cells, so naive T cells are essentially like a newborn baby, right? They don't know what they're going to be. They don't have you can't tell what personality they are going to end up having. And after an infection or something that initiates an immune response, those T cells can differentiate into different, functional groups, right? So they're in the end, they're not all the same. And once they do that, that process involves a lot of chromatin modifications and epigenetic changes, which open up loci for certain effector functions or cytokines and not others. And so that T cell will now always make interferon every time it gets activated, but not make IL-4 or some other cytokines, whereas other T cells will make, have different functions. And so I think the signaling at the level of the membrane and the components I talked about, they don't change, but epigenetically, the response of the T cell can be very, very different because it's got open loci for some things and closed loci for other things. Mm-hmm. That's how it's generally. I think there are differences between CD4 and CD8 T cells. I don't think it's differences in the proteins involved in the signaling. It's more about their sensitivity and ability to get activated.
Right.
There was one question about that there are inhibitory and activating co-stimulatory receptors in the CD28 family of molecules, surface molecules. And the question is, how does the downstream pathway differ between co-stimulatory and inhibitory CD28 family receptors? Maybe you can clarify that again.
Yeah, so I think that the answer is that it's kind of interesting. Both CD28, which is a co-stimulator, positive regulator, and one of the negative regulators. Let's talk about PD-1, for example. They have cytoplasmic tails that get tyrosine phosphorylated, but the sequence of amino acids around that tyrosine differs between the positive and the inhibitory receptors. So with the positive receptor recruits, like CD28, when it's phosphorylated, it recruits positive signaling proteins like the regulatory subunit of PI3 kinase or adapter proteins like GRAT2 or kinases like LCK. And the inhibitory receptor PD-1 the amino acid sequence around its tyrosine that gets phosphorylated recruits SH2 containing phosphatases. So it's all about not just about the receptor itself, but about what proteins it recruits or get recruited to its signaling tail when it's phosphorylated. So if you recruit a phosphatase and activate that phosphatase, it's going to undo all the good deeds of the antigen receptor and the CD28.
Yeah, thank you for clarifying that. I think that's an important piece of information. Another question maybe related to that is that regulatory T cells that inhibit the function of conventional T cells, can they actually engage these inhibitory pathways to modulate immune activation?
They do. Well, they do it indirectly. So one of the mechanisms by which regulatory T cells shut off other T cells is by expressing a protein called CTLA4, which is a CD28 family member. CTLA4 actually binds to the same ligands on the dendritic cell that are needed to activate the T cell that normally bind to CD28. And turns out that CTLA4 binds with much, much higher affinity to those ligands on the dendritic cell. So when a regulatory T cell, which constitutively expresses lots of CTLA4, shows up, its CTLA4 will bind up all the ligands on the dendritic cell, depriving the T cell trying to get activated from having the ability to engage its CD28 co-stimulatory receptor with those ligands. So it's kind of an indirect way of the inhibitory receptors blocking a co-stimulatory receptor. It's not a direct effect of them interacting.
Okay, then there's also one question related to a molecule that you have not talked about, and I'm not sure if, whether you can comment on that. And that's, I think, it's related to the alpha-7 nicotinic acetylcholine receptor, which was shown to be an anti-inflammatory pathway. And I'm not sure whether this is in T cells or other immune cells. And the question is, does this pathway actually reverse these activator signals, for instance, tyrosine phosphorylation that you have talked about, or would that involve other modulation of other signaling pathways?
Yeah, I mean, I honestly have no idea, but I doubt. So my understanding, okay, this is I'm going to embarrass myself here. Rudimentary understanding is those receptors are very unlikely to involve tyrosine phosphorylation as a signaling mechanism. Mm-hmm. I could be wrong about that, but if they activate G proteins, heterotrimeric G proteins or something, then that's a very different mechanism of signaling. And so most of the inhibitory receptors in T cells at least work by somehow undoing tyrosine phosphorylation. And I would suspect that it would likely be a different mechanism.
I was also intrigued by your picture that you showed about naive T cells, the electron microscopy pictures about naive T cells and the effector T cells, and how the sheer size of the cytoplasm changes, right? And when we draw these pictures of signal transduction in immune cells, it's always this empty space and there are molecules floating around in kind of empty space, and it's a long distance signaling. But as you pointed out, in naive cells, this all happens in very small microdomains. And I wanted to use this picture maybe as a transition to our next speaker, Rich Lewis, who will talk about the mechanisms of calcium signaling, especially stim-ORI signaling, which happens in these ER plasma membrane junctions. And maybe one question, and asking your opinion on this too, Leslie, is, how does the structure, the morphology of this of the cytosol of the cell affect signaling, especially if you compare naive and activated cells, not just protein expression, but also the sheer difference in the structure of cells. Is that well explored, or do you think this needs more in general, more work?
I think Rich certainly might have something more intelligent to say about this than I do, but I know, there are people trying to study that these, there are thought to be, for example, on the membrane, like protein islands, right, where there are places where proteins are collected and other areas of the membrane that are relatively sparse in proteins. And I think more and more, signaling pathways, and this is true for transcription factors and transcriptional activation, are thought to happen in these condensates, right? Where proteins are not actually just floating around in solution, but they're in these they're concentrated into very small volumes where these interactions that we draw out in these cartoons, are much more favored. And so I would guess that the cytoplasm also has these condensates of signaling modules, like these, the signalosomes people talk about, right? Where LAT has got the adapter protein, has all of these other proteins bound to it. I suspect that those happen in these condensates, but
yeah,
definitely needs more exploration.
Great, thank you very much.